Okay, so we stopped with the important step, last one, where we understood that the primer binding will have an extension of 4 nucleotide which can have combination 256 different combinations. Why we require 256? Because we do not know what is the sequence of mRNA that we have isolated. So if we use all the 256, there is a possibility that at least one will 100% bind to the mRNA and extension in the PCR extension step will be carried out and your PCR product will be produced. Now, depending on the number of times a particular product is produced, number of times the fluorescence is produced, you can tell how many mRNA molecules are present in the given pool of sample that you have given. So, this method is called as TOGA, where there are 256 reactions performed with fluorescently labeled 3' tags in order to generate fluorescent PCR product. And to identify the expressed sequences, a digital address is created for each transcript identified. That means whatever primers you are designing, similarly your software will also contain the address. That means the same sequence will be present. And once the TOGA experiment starts, whenever there is a fluorescent tag product is being produced, real time it is done in real time. We have studied what is real time PCR. It is connected with the software and you have those fluorescent primers which will help us in understanding in each reaction how many products are produced. So at the end of every cycle you will know what type of products are produced. So based on the database available, you can identify or address out of the different combination which one has given the maximum number of sequence data. And these sequence tags which are produced, they are called as, okay, now here one more thing which you need to know is, when you are amplifying, one second. So initially we saw that the entire mRNA was converted to a cDNA, and in this cDNA we cleaved it with the restriction enzyme and we have taken only this portion for our next step. Now depending on this position of the restriction site, for example this is one mRNA, this is cDNA, in one of it was present here, in the second one it was present here, in the third one it was present here, in another one it was present here. First restriction site, I am not talking about all this, I am talking about only the first restriction site. So now you can see the length of each cDNA that you have in your hand, right? And this is what is used with the anchor sequence, 8 nucleotide sequence. This is what is amplified. Now this length, right, this length and this sequence of the parsing sequence, these two will help us in uniquely identify each mRNA, this and this, right? So that is what I am mentioning here. Since the cDNA was originally cleaved at a discrete site upstream of the poly A tail, that is on the left hand side of the poly A tail, when the length is included, which is what do you mean by length is included? From the site of restriction to the poly A tail, that length what I showed you just now, that when that length is included, an address for each RNA is obtained, right? This is called as an address of each RNA. So this is what we are going to use to uniquely distinguish two different RNA population, and these are called as digital sequence tags. Please remember, these address or the identity of the 8 nucleotide plus the length of the fragment, that is sufficient to recognize an RNA if its sequence has already been deposited in the database. This is called as digital sequence tags. Okay, so using this technique you can identify different types of RNA present in a population. And usually TOGA is not carried out only with one type of restriction enzyme. Usually 4 different restriction enzymes, each one containing a unique 4 nucleotide, 4 base pair recognition sequence is used. If not for one, for the if an mRNA does not have restriction site for one restriction enzyme, it might have it for the other restriction enzyme. That is why 4 different recognition restriction enzymes are used. If we use only one restriction enzyme, there is a possibility that one particular mRNA which is present in your cell might not have any restriction site for that particular restriction enzyme. So in that case, you will never be able to address that mRNA at all. So in order to overcome that problem, scientists use 4 different restriction enzymes with each of the mRNA. Some of them might get cleaved with all the 4, some of them will not get cleaved, some of them might get cleaved by only one. And also remember, all the 4 restriction enzymes are not used simultaneously. All the 4 are used separately in 4 different reactions. And then later, once TOGA has been completed, automatically it will compare the results of all the 4 restriction sites, restriction enzyme data, and you will get a graphical representation of the expression of various genes, which mRNA was produced, how much it was produced in certain cells, whether that mRNA is produced or not. In certain it might not be produced, in certain it might be produced. So this is what we understand by TOGA. And one of the main advantage, as I have already told you, is very low amount of starting material is required and you get very good result, which is repeatable and can be easily understood. So with that we complete TOGA. Thank you.